Okay, well, thank you everyone for coming. And as this is technically my first time hosting a speaker, it's definitely probably my fault that I didn't do the time frame and help with that process. So I will take some of that responsibility for sure. Today we have Dr. Rob here. He is an assistant professor in the plant biology section of the School of Integrated Plant Sciences at the University of Cornell. He obtained his PhD in genetics and quantitative biology from the Shu Lab at Michigan State University in 2013. Following that, he continued with a postdoc also at MSU and Dr. Robert Last's lab. And now his lab at Cornell works on understanding the evolution of plant metabolic networks. And this is from the molecular to ecological scale. So today his talk is entitled Evolution of Diversity of Defense Sugar Metabolites in Solanaceae and the, yeah. And so now I will yield the floor to our speaker.
Okay. Can you all see my slideshow?
Yeah, that's perfect.
Okay. All right. Again, 1,000 apologies for starting late. I will thank all of you, not just the organizers right now, for waiting to hear my talk. I'm going to cheat a little bit and also talk about Convolvulaceae. It's the sister family of Solanaceae in here. So just to orient where I'm located right now, I'm talking from Ithaca, which is about 5 hours away from New York City in upstate New York. It's really a beautiful place, but just 10 days ago, this was how Ithaca was. There are below. This was my front yard, and it was covered with snow just a few days ago. And today the temperature is 4 degrees Celsius. So all of this has melted within the last week. It's not always this bad. You also in fall and summer, it's actually pretty beautiful. And if you get a chance, please do come here in upstate New York. So my lab works on evolution of plant metabolism. I'm basically curious about how metabolic diversity has evolved over time. And there is still a lot to know. Despite our knowledge of metabolic pathways, we still have a lot to be discovered at different scales. So my lab works on looking at different scales of at which metabolic diversity and plant has evolved. So we look at molecular scales, looking at evolution of proteins, as well as at ecological scales and evolutionary scales. So in this talk, I'm going to first give a short introduction to what defensive sugar metabolites are and why I'm interested in them, and then talk about the diversity of these metabolites in Solanaceae and Convolvulaceae. And finally, I will spend some part of my talk on our recent study in Solanum habrochaites. So in the most well-studied defensive sugar metabolite, arguably, is this class of compounds called acylsugars, which is restricted to the Solanaceae family. So acylsugars comprise of this sucrose core, to which there are different fatty acids attached. But in the sister family Convolvulaceae, we also have a similar type of compounds. We have something called resin glycosides, which have an oligosaccharide core, to which there is this long acyl chain of C16 hydroxylated fatty acid attached. And then there are other acyl decorations too. But this is not just restricted to Solanales lineages. It's also present. These type of compounds, not exactly the same, but similar type of compounds have also been reported in geranium, in Caryophyllus species, and in other lineages too. So either it's convergent evolution in different lineages, or or there is independent evolution, or perhaps there was an ancestral state, and these compounds are very ill documented if it was an ancestral state. The common feature of these compounds is having a sugar backbone highlighted in blue, and some acyl chains, either red or black, shown in the figures here. Okay, in Solanaceae, these compounds have been studied very well. So these acyl sugars are produced from this tip cell on top of trichomes in different Solanaceae species. And these are important for deterrence of oviposition by thrips, by whiteflies, etc. Ian Baldwin's lab has also shown that these acyl sugars mediate tritrophic interactions. So these hornworms, which feed on Nicotiana leaves, they ingest the acyl sugars, and the acyl chains are cleaved in their hydrolyzed in their guts, and these cause the caterpillar to the larvae to smell because these are volatile, and the smell, the volatile acyl chains are then detected by ants or moths, which come and eat these larvae. So it provides some sort of a tritrophic defense to the larva. In resin glycosides have also been studied for quite some time from a chemical perspective. So these are known to be active against a wide range of living organisms. These have also been components of plants used in traditional medicine. So resin glycosides are, Ipomia purga is used in Mexico as a purgative, and similarly Operculata terpethum, which is also a member of the Convolvulaceae family, that's used in India as a purgative. Sweet potatoes, which are members of the Convolvulaceae family, uh, sweet potatoes that produce more resin glycosides are known to be affected or less affected by nematode rot and insect damage compared to other sweet potatoes. So these have biological effects. We know they are defensive, they are produced for some reason by these plants, but their ecological role is not known. Now, I was interested in looking at the diversity of these compounds. So initially this project, which is published in eLife in 2017, was started when I was a postdoc in Rob Last's lab at Michigan State. This project started off with us going to the New York Botanical Gardens and sampling about 40 different species from the Solanaceae family. And essentially we did LCMS. So in that case, you basically take a leaf from one of these species, put it in an organic solvent, and extract out all the surface metabolites, not even grinding it. It's just extracting the surface metabolites and running it on an LCMS. So the profiles that we get look like this, and almost all of these are acyl sugars. In some species, we see over 900 different acyl sugar-like signals. So why do these species produce so many acyl sugars is not known. Why not just produce 20 or 30 acyl sugars abundantly? So one of the important things to note is that a lot of acyl sugars make the surface of the plant very sticky, and that's what causes the oviposition deterrence in for some species, for some insects. Now we can delve down deeper by looking at the mass spec data and try to predict what sort of acyl sugars are actually present. So typically we see a sucrose core to which these fatty acids are attached, but we were also able to find some species like Solanum nigrum, Solanum ketoense, which instead of sucrose use glucose or inositol. Solanum ketoense is the only species that we found that uses inositol for making these acyl sugars. And then the acyl chains can also vary. They can change from C2 2 carbon long to 12 carbon long, and they can be on different hydroxyls. So there is quite a bit of variation that produces that exists in the acyl sugar phenotype. Now, what causes this diversity at the biochemical mechanistic level? In tomato, so this is all the 4 enzymes that are involved in the production of these acyl sucroses are members of this family called the BAHD acyl transferase family. In tomato, there are 4 enzymes which are known to carry out these reactions. But in other species like Salpiglossis sinuata, this is one of the early emerging extant plants from one of the early emerging lineages in the Solanaceae family. And in our study, we found that there are at least 5 enzymes in this species, all belonging to the same family. And they have been involved in, they have a lot of promiscuous reactions, which ends up producing a lot of structural diversity of acyl sugars. So in that study, we were also able to figure out that the ancestral state of this pathway involved 3 enzymes, which we call acyl sugar, acyl transferase, or ASAT1. So there were 3 enzymes at least at the common ancestor of all these species and of all Solanaceae species. This phenotype does not exist in Convolvulaceae, only in Solanaceae. So at this point, in most of the Solanaceae, you find orthologs of these 3 enzymes, ASAT1, 2, and 3. But only in the Solanum genus, the ASAT1 is completely absent. So the red ASAT1 is completely absent. Instead, what we found was the ASAT2 and ASAT3 have actually shifted their activities to become ASAT1 and ASAT2. So how exactly this happened, that's still being researched in Rob's lab. But this was an interesting finding that there's a loss of ASAT1 and switching of activities. It probably occurred because of existence of promiscuity in ASAT2 and ASAT3 for using alternative substrates. Now, for this enzyme family, actually, we have been, after I started my lab, I started looking at this enzyme family in more detail, not just acyl sugars. And we have this study in bioarchive that's currently under review, which looks at the whole evolution of this family across the plant kingdom. And if you are interested, this would be definitely a paper to look at. Right, moving on to Convolvulaceae resin glycosides now. So chemically, a lot of structures have been characterized using NMR for these compounds. So we know there exists a lot of structural diversity, but it was not put in the phylogenetic context. The biosynthesis of these compounds is not known. And because the enzymes are not known, we cannot say why they are not produced in Solanaceae. Is there an independent evolution of enzymes that occurred in the Convolvulaceae family, or were those enzymes lost in Solanaceae? We cannot say that. So when we started off this project, we again utilized a similar approach by looking at different species of the morning glory Convolvulaceae family. And again, we see a lot of diversity here. The x-axis, this is the chromatogram of extracted ion chromatogram actually for different species. And the x-axis is retention time, y-axis is the ion abundance. And here you see that the resin glycosides that are obtained are shifted. They are not exactly at the same location. And hence, there is some diversity that exists here too, which we wanted to probe in more detail. Getting a foundation of what chemical structures exist will help in further elucidating the biosynthetic pathway. So if you go deeper into each of these peaks, you can get the fragmentation patterns of these peaks. So in this case, you have in this case, we knew the structure of this compound, tricolorin A, which has a mass of 1022.5662. And then when you look at the fragmentation patterns of this compound, you see these particular peaks. Based on the knowledge of these structures, you can associate it back to what the original structure, what these fragments would be. In most cases, we don't know what the original compound is. We only know what the fragments are. So an analysis of these fragments can help us piece together what chemical substructures exist in different resin glycosides. So we've done something of that sort. We looked at about 30 different species of the Convolvulaceae family. and looked at root and leaf phenotypes, resin glycoside phenotypes, and the different acyl chain fragments, etc. So some of the striking patterns that we see is Convolvulaceae and Callistegia species here produce a lot of resin glycosides. We don't know exactly why that's happening, or whether this is unique to Convolvulaceae. We do see some high number of peaks in Hypomia species too right here, but uniformly these many of these convolvulaceous species show or convolvulus species show a high number of resin glycosides. We also see that most of the time the resin glycosides are produced in the root. So if you take all the resin glycosides and ask if they can be explained by the resin glycosides present in the root or the leaves, we see that in almost all cases, if a resin glycoside is present in the root, it's present in the leaves. So we know that they are produced in the roots. Whether they are produced in the leaves, we don't know that yet, because when they could also be transported from the roots to the leaves. But in this case, there is one species that threw us off, Convolvulus tricolor. In that case, all of the unique resin glycosides are produced only in the leaves and not in the roots. So whether they are produced in the roots is still, in the leaves is still a question. We also looked at various different the long acyl fragments existing in resin glycoside structures, and 260, this particular mass, 271, corresponds to the C16 hydroxylated fatty acid mass. We also looked at C14, C16, C18, and so on, but we could not detect a lot of those compounds. There exists some compounds with those fatty acid chains, but not a lot. And I'm not showing this, but in most cases, we were able to show that these C16 hydroxylated fatty acids are attached to deoxyhexose instead of hexose or pentose sugars. No, all of this is based on MSMS fragmentation patterns. So we wanted to first verify that these actually are compound resin glycoside structures. So we purified a couple of compounds. I'm showing one structure here, purified one of them and looked at and did an NMR. And we were able to confirm that in Dichondra argentea, this abundant peak that we see in mass spec was actually a resin glycoside. So that was good. This plot is molecular networking of resin glycoside-like structures. So essentially, you take all the MSMS fragmentation patterns of resin glycosides and associate them in a network fashion. So this particular view shows that there are many. So each node here is colored by. Each node here corresponds to a unique resin glycoside profile, and the colors here are based on the different genera. And we see here that Convolvulus and Hypomia produce many resin glycosides that are unique to those genera and do not exist anywhere else. But there are many resin glycosides which are very similar, and even though they are produced in individual species, like this yellow dot right in the center would be produced by Callistegia, but it is structurally very similar to other resin glycosides produced in other species too. This particular region is sort of a common substructure that exists in all of the Convolvulaceae. The unique resin glycosides produced here too. Then we can dig further into the profiles and mass spec data and ask what are these unique compounds. In Convolvulus, it is quite clear. So here I'm separating all of the signals by genera again and asking which particular peaks are present only in one genus. And this particular group is present in only Convolvulus, and these are very high molecular weight compounds, over 1500 M by Z. And this essentially is indicative that Convolvulus produces very large resin glycoside structures that are not seen in other species. So the next question then is, how can we use these resin glycosides for understanding their biosynthesis? And this is still an ongoing work. We are doing proteomics, developing transformation assays in convolvulaceae species to figure out the different enzymes that might be involved in their biosynthesis. But a common theme here is, again, we have large enzyme families like glycosyltransferases, P450s, acyltransferases involved in the biosynthesis, and large enzyme families are known to exhibit promiscuity, and it's likely because of this promiscuity that you are getting so many diverse structures, 300 signals in one species, in a leaf of one species, and that is likely because of the involvement of these large enzyme families. Okay, summarize this part of the talk. I've talked about how resin glycosides are very diverse metabolites in the morning glory family. They are made in roots, and whether they are made in leaves or simply transported is not clear. We see evidence of lineage-specific innovation in convolvulus and in hypomia, and we need to know the enzymes to figure out why that has happened. Um, and this particular study was also an example of how we can use different informatic methods. Like we've analyzed quite a bit of the data using custom Python scripts that we wrote in the lab, and using various different cheminformatic softwares. So using that too, you can dive deep into LCMS data and get a lot of interesting evolutionary insights that wouldn't be just possible by looking only at a narrow subset of the phenotypes. Right, so I'm going to switch over a little bit and talk about acyl sugar diversity. I don't know how much time I have, 15 minutes. So in Solanum habrochaites. So this was a study that we recently, it's under a review right now. But these acyl sugars are produced again on the trichomes of Solanum habrochaites, which is closely related to Solanum pennellii and Solanum lycopersicum. Solanum habrochaites actually is present in Peru and Ecuador. It's present along the Andes mountains, and the red, which they are shown, habrochaites accessions are shown here in blue, and Solanum pennellii, its sister species, is shown in red here. A previous study found out that in tomato, you only have about 8 or 9 different acyl sugars. They have probably been bred out, but in Solanum habrochaites, you have over 120 different acyl sugars, which makes this plant very sticky. If you're working with it in the greenhouse, your hands will get all sticky, and same is true with Solanum pennellii. So a previous study in Rob Last's lab had looked at various accessions of Solanum habrochaites spread across the range, and they found 2 main superclusters of these acyl sugars. And so this is arranged from south to north, and in the northern part of the range, essentially, there is a loss of acetylation. So there is an enzyme. Yeah, so if you look at the accessions across the range, then in the southern part of the range, you get fully formed acyl sugars. But in the northern part of the range, they found that one of the enzymes, the 4th enzyme in the pathway, the last enzyme in the pathway, it has been inactivated through different means, producing these green, orange, and blue chemotypes. So this is not just in acyl sugars, even in terpenes, there was a pattern found in terpene structures from Solanum habrochaites trichomes, which, and in the northern part of the range, there was more diversity. So when I started off this project in Rob Last's lab, initially, I was curious about how population structure and movement of Solanum habrochaites across the range has contributed to evolution of these phenotypes. So when I started here at Cornell, we continued the study with Jacob Landis here, who is a postdoc in Chelsea Specs Lab. Um, and we actually built up a collaboration working with Pat Bedinger's lab, Pat and Amanda at Colorado State. And it's been such a wonderful collaboration, because initially we started off only thinking about population history, but when reproductive phenotypes were combined with the study, it led to a whole new level of evolutionary insights. And Pat and Amanda will be talking here in Solanaceae seminar series in April. So this paper is currently under review if you're more interested in the story. And the initial idea was to sequence different habrochaites accessions and characterize the population structure and gene flow and associate the patterns with acyl sugar diversity. So initially, after doing the RAD-seq, I won't go into the RAD-seq details, but the first thing that stood out was that some accessions here, these accessions are organized from north to the south, and some accessions really stood out. These accessions, when you look at the acyl sugar profiles, they had also stood out back in the 2012 study. So this orange accession should not be here, it should be on top. And similarly, the orange accession here should be in the northern part of the range, and the purple accession area 2650 should be in the southern part of the range. So that was heartening that our our RAD-seq profiles actually matched what was seen in the chemotypic patterns. When we looked at the principal component analysis, it led to a different insight. So here, these two populations, the purple and the red, are right next to each other. So this is the purple population, and this is the red population, and these are all purples. They're right next to each other, but at the genetic level, they are very, very distinct. So purples are actually more related to populations here, in the accessions here, than this population. So that suggests that there is some demographic, it wasn't a straightforward migration of Solanum habrochaites. When we looked at the phylogenetic relationships between these accessions, it was we were able to classify them, all of these accessions, into 8 different population clusters. Interestingly, there's a big difference between 2 groups that we call supercluster 1 and supercluster 2, and that occurs right at a region called the Huancabamba Depression. And this whole region is called Amotape Huancabamba Zone, and it has created a lot of variation. So you see there are only 2 main clusters completely south of the AHZ, but there are 6 clusters within the AHZ and to the north. So something happened in the AHZ which led to production of generation of these many clusters. So looking at AHZ, it's really a fascinating region. This is a region where the Andes Mountains shift their directions and move further down south along South America. And this is one of the most biodiverse regions in the world. It has about 1/6 of plant life, not the AHZ, but the whole tropical Andes ecoregion has about 1/6 of plant life. But AHZ itself has a high degree of endemism. This is one of the lowest lying areas and actually creates corridors for plant movement from west to east and east to west. This is a region that's of interest to us. Now, Solanum habrochaites too, this is one of the 3 Solanum species in the Lycopersicon section that's found both north and south of the AHZ and within. So Solanum habrochaites here, accessions are seen in H, Pimpinelli folium is seen in P, and then Solanum neoricae is seen in red. Some of these are arcanum, but you can see neoricae in the AHZ and to the south. So Solanum habrochaites managed to traverse the entire AHZ and move to the north and south. But it wasn't without any effect. So this region already has high endemism. And when you look at so Pat Bedinger's lab looked at different reproductive phenotypes, and they found out that there is a lot of transition from an ancestrally self-incompatible populations to various independent emergence of self-compatibility in the northern part of the range. So there is a lot of reproductive isolation occurring due to geographical landforms. There are deep valleys and high mountains in this region, which creates local differentiation and reproductive isolation, and that has led to emergence, or is associated with the emergence of self-compatibility groups north. In the south, again, these are very distant accessions compared to the others, and here too we had an independent emergence of self-compatibility down in the south, that is denoted as SC4 group. And again, as I said, Pat will be giving a talk on April 30th, I presume, and you're encouraged to come join again for more detailed results on this and other of their projects. Now, when we looked at using RAD-seq data, we can look at heterozygosity patterns associated with self-compatibility and incompatibility in different regions. And we find that these self-incompatible populations have a bit high heterozygosity, as we would expect, compared to self-compatible populations in northeastern Loja and southern Peru. When we look at how gene flow is occurring using FST, interestingly, the self-compatible populations again are quite separate from self-incompatible ones. They show longer branch lengths here, but cluster 4 and 5, which are both self-incompatible, also are, and they are right next to each other. They are very close to each other, but they lie in the AHZ. This is like they are separated by the Huancabamba depression, and cluster 4 especially appears to have a high local differentiation and reduced gene flow compared to the other clusters. Now, when we look at the self-compatibility mappings and population clusters, they explain the acylsugar phenotypes. Starting from the north, where so this is the population where ASAT4, well, I'll start from here, right in the middle. These green triangles represent ASAT4s that the gene is present. You can detect the sequence, but it's not expressed. The orange ones are the ones where the gene is expressed, but there is a frameshift mutation that causes functional inactivation. And in the 4, the PCR primers couldn't even bind. So we don't know if the gene is highly differentiated or if it's just lost or deleted in this population. So there are 3 different mechanisms by which ASAT4 was inactivated in the northern part of the range. So we see here that these purples, the blues are associated with an independently emerged self-compatible group in the northernmost part of the range. This entire set of orange dots is associated with emergence of SC1 self-compatibility group in northeastern Loja, and these individuals also hybridize with other individuals up here, which results in production of these orange dots in this part. And then finally, this particular group, the green one, where the gene is present but it's not expressed, all of those are self-incompatible populations. But I'll come to that in the next slide. This is cluster 7, the highly differentiated group that we saw. It also has a unique chemotype in acylsugar profiles. And in the same, this stands out as a very different population from the rest of the Solanum habrochaites populations at the genetic level using RAD-seq data. The SC4 group, which is cluster 8 at the very south, it's doing its own thing. Now, one of the interesting observations was that this particular group, it's all self-incompatible populations, but still some of them have an active, the blue ones are the ones with active ASAT4, and the green ones are the ones with inactive ASAT4. The expression is lost. When you look at how Solanum habrochaites has moved through its range, we see that most of the time it is on the western slopes of the Andes Mountains here. But then at this point, it moves into the Andes and it stays in the Andes. So this region actually corresponds to the southern boundary of the AHZ, the low-lying areas through which habrochaites was likely, it found a path to enter the Andes Mountains. And this is exactly where this region actually maps very closely to where the inactivation of ASAT4 has occurred. So I'm not saying it's a cause movement into the AHC was the cause of inactivation, but it's certainly associated with inactivation of ASAT4 and emergence of a new acyl sugar phenotype. So based on our RAD-seq data and all, as well as the SCSR definitions, as well as the acyl sugar phenotypes, we were able to come up with a model for how Solanum habrochaites emerged. This region has been predicted to be the origin of Solanum habrochaites for a long time, and our results confirm that finding. After origin, it moved. There were two branches. One moved to the north, one moved to the south. But there were independent establishments of cluster 7 here in the Ancash region, as well as clusters 5 and 6. And then there were independent evolution of compatible groups in the northern and southern part of the range. Right. So just a couple of slides remaining. One is, here in this part of the study, I hope I have shown that there is very interesting biology occurring in Solanum habrochaites, which has still been understudied. We looked at only two phenotypes, acyl sugars and reproductive phenotypes, but also terpenes. But there will be several more phenotypes that have been altered through the movement, because of the movement through the AHZ, which needs to be studied more. And then these isolating geographic features led to emergence or associated with emergence of self-compatibility, which are then associated with evolution of new phenotypes. So it all ended up as a nicely tied up story at the very end. So to take some take-home messages from my talk, the first, before everything else, is know your time zones and don't be late for talks. Second is that there's huge amount of plant biochemical diversity, and it's really understudied. With LCMS, with new computational methods, we are able to study and probe into this diversity very well. And this diversity is associated with further evolutionary and ecological questions. Yeah. So before I end my talk, I just wanted to make another plug. So Stacy Smith, me, and a few others are involved with starting this new phytochemistry section at the Botanical Society of America. This will be initiated from the Botany 2021 meeting. We have a colloquium titled Phytochemistry from Atoms to Ecosystems, where we have a very nice lineup of speakers already from various different fields: ethnobotany, how plant chemicals affect gut flora and our physiology, chemical ecology, lipid biosynthesis, chemodiversity, evolution. So please do register for Botany 2021, and please do attend this session if you like phytochemistry, doing phytochemistry research. Please join us in this section. We are also organizing a separate session from abstract. So if you have some biochemical research in your lab, if you are doing something, please do submit these abstracts to the section when registration opens. And this meeting is virtual, so international participation, even when it is on site, it's very welcome. So please do register for Botany 2021. Right. A lot of people helped with this study. I'm not going to mention everyone. It's a collaborative work across different labs, but I would especially like to point out the work that collections do in facilitating projects like these. And yeah, I can take any questions.
Thank you very much, Gaurav. That was amazing. Super great talk. I think Corey is here and she's going to moderate the questions.
Okay.
Yeah, so if you have any questions, go ahead and put them in the chat. Would you like me to say this question? Or do you want to unmute yourself?
Yeah, there is a question about whether the changes in Solanum habrochaites are just seen in the chemical profile, or if there is a change in the morphology of the trichomes, like no glandular ones. No, the glandular there are Solanaceae species have type 1, type 4, type 6 trichomes. Type 6 trichomes produce terpenes, type 4 trichomes produce acylsugars, flavonoids, etc. So there is no uh, we see terpenes, type 6 and type 4 trichomes are present everywhere, just the profile changes. But we've seen, when growing these plants in the greenhouse, I've seen the leaf morphology being different. We haven't really looked at trichome density very closely. If that's changing, we know that the investment in constitutive versus inducible defense is altering through other research that's going on elsewhere. So things have changed at the phenotype level.
Thank you. Yeah, now I think there is a question of Lucio D'Andrea that he wants to unmute himself and the question.
Yes. Hello. Very nice talk. I really enjoyed it. Can you hear me properly?
Yeah, I can hear you.
Okay, great. So I have three main questions, let's say, if I can. So one is like putting together the information that you gather from Solanum nigrum in the first project from the eLife in 2017, and those things that you are observing now in Solanum in Peru and Ecuador. So I was, because Solanum nigrum has a different sugar core, right? I was wondering if that could be a sign of local adaptation, because nigrum is an invasive species and is known to be a polyploid coming from Villosum. So I was wondering if might be something related with that.
Yeah, it is very much possible. We got Solanum nigrum at the end of the day of our sampling. It was not even in the New York Botanical Garden collections. It was growing by the roadside, actually, when we were driving around. And Rachel Myers was helping us with the sampling, actually said, oh, that's Solanum nigrum. So it
Okay.
could be an invasive one. We did a flow cytometry. It had a high genome size. Now, whether it's caused due to local adaptation, I don't know. If it's adapted or it's just due to genetic drift or it's something that's more ancestral. I did look at some other related species. There was a mix of acyl glucoses and acyl sucroses in I don't remember the names of those species, but it's not just in Solanum nigrum. The related species, I think Solanum americanum also shows some bit of
Villosum. Yeah, Villosum americanum. Yeah.
Yeah, it also shows some acyl glucoses. These are produced by an invertase, which make the acyl sucrose is made, but there is a cell wall dependent invertase which cuts the glycosidic linkage, producing the acyl glucoses. This happens in Solanum pennellii too. So Solanum pennellii produces a mix of acyl sucrose and glucose.
Okay. Okay. Very interesting. Thank you.
Yeah.
The second, if I can, if the host, yeah, okay. So the second question is about the plasticity. So you, metabolic plasticity, I mean. So you are observing that there's an increase in the metabolic diversity sometimes, and we don't really know what those acyl sugars actually are doing. So do you think that plants are accumulating different acyl sugars? Those acyl sugars are doing something different physiologically, or are they plastic? Let's say that they are doing the same thing, different molecules.
This has been a controversial question. I come from a neutralist perspective. I tend to think that these are not doing anything different, except increasing the stickiness on the surface of the plant. Um, there is and the inherent reason why so many are being produced is just because of the promiscuity or the broad substrate specificities of the underlying acyl sugar acyl transferases. Having said that, there is some evidence that acyl chains do have an effect on, Ian Baldwin's work has shown that ants that grow near plants which have a C6 chain attached to them are more attuned to smelling that smell versus ants that are near the Datura plants which produce a C8 or a C5. They don't get attracted to the C6 signals as much as C5. So there is some difference, but I don't know if that can be generalized to every acyl chain having a functional role.
Okay, great. And the last one is about if you calculate the Shannon entropy for the morning glory, using the morning glory peaks from the MS.
We have done that. We have done Shannon entropy calculations. I don't have the result. I don't remember the result. We did it some time ago.
Okay.
Yeah, I cannot comment on that.
Okay. Thank you. Thank you a lot. Thanks again.
Thank you. Federico?
Hi. Yes, great talk. Very interesting. So I have two questions. One is, between the convolvulaceae and the Solanaceae, how different is the biochemical pathway in terms of the enzymes used? Are they homologous or autologous? You don't know? Between the two families.
Yeah, so for acyl sugar enzymes, in the sequence genomes, we could not find their orthologs in any of the Convolvulaceae species. And we don't know the enzymes for resin glycoside, so we can't see in subnation. And
the other, yeah, great, thanks. It's about also related to the plasticity and the environment and the genetics. So did you find a strong relationship, for example, in Solanum habrochaites, or when you did study several species, between genetic variation and metabolic variation? So normally, if you cluster the species by the metabolomes in Solanum habrochaites, they also cluster in the same way as genetically, or not necessarily?
In terms of, yes, I would say yes, they do cluster. In this figure, actually, we see that there is quite a bit of relatedness between. So this is all chemical clustering on the left, and the right is is genotypic clustering. So the pie charts are actually the percentage genotype based on RAD-seq and targeted sequencing data.
Okay, thank you very much.
Yeah.
I don't see any more in the chat, but does anyone else have any more questions?
Funky hydroxyacyl chains are being made. I know the hypothesis is P450. How they are made, we've done some feeding experiments now with palmitic acid, C16 fatty acid. Our LCMS broke down recently, so we are trying to, when that is fixed, we'll have a better understanding of how they are being made. All right. Thank you all for waiting, and thanks for.
Yes, thank you very much, Gaurav. That was an amazing talk, and thank you for the questions as well. I think if there are no more questions, we are closing the seminar. But of course, that all the attendees, I can send you questions through email, or we're going to send you the chat as well.
Yeah, good. Thank you.